I'm very excited to give you a presentation on my PhD thesis topic, which is titled Contributions to Image-Based High-Throughput Phenotyping in Viticulture. And yeah, I think we just dive into. So first, I want to answer the question, why is viticulture an interesting field to study? So although you can see this is the whole area of arable land in Germany, horticulture, including viticulture, only makes up 1% of the arable land in Germany. So why is it interesting to have done research here or to investigate how to make this part more sustainable? The reason is that in grapevine farming, the use of pesticides and fungicides is really high. So although only around 1% of the area is covered by vineyards, nearly 20% of the pesticide and fungicides are sprayed here. So if we can make an influence in this small part of the arable land in Germany, we can actually make a great difference in the amount of applied pesticides throughout Germany. But at the same time, we have to keep in mind that in contrast to many other crops where we have the problem of feeding the world, we want to maximize our yield. In viticulture, it's quality over quantity. So here we have a fixed amount of nutrients which are available to the plants. So the nutrients have to be shared. And to increase the quality, we even sometimes have to remove some of the yield components. And in Germany, this is actually regulated. So we have some different quality names for wine in Germany. And the highest one, the quality wine, has the restriction of just 100 to 125 hectoliters per hectare. So although we want to improve the sustainability of grapevine farming, we also have to keep in mind that we still have to meet the quality standards. So how can we actually influence the quality or also the sustainability? I think for all of you here, I don't have to tell you too much about phenotyping, but nonetheless, a quick reintroduction. So the phenotype is actually what we see in the field, and it consists on the one hand of the genotype, so which plant is actually planted in the field. And then, of course, the environment influences how the plant is growing. So if we have really dry weather, the plant is developing differently to very humid weather or a lot of rain. And we can also influence the growth of the plants by management decisions. So here we can say, do we want to remove these yield components or defoliate the plant to let more sunlight hit the grapes? But these are things how we can influence the phenotype. So in viticulture, phenotyping is a very labor-intensive and manual task due to the perennial nature of the plant and also to the often challenging terrain situations. So usually what is happening, we have the sampling step. So a farmer or a breeder is running through the vineyard and is looking at single plants throughout the vineyard. These are then investigated in detail, and in the end, some decisions are done based on the extrapolated data. So here we could, for example, investigate the expected yield at the end of the season, possible diseases which affect the plants, or also see if pests are apparent in the vineyard. In the following, I will go a little bit more into detail about yield estimation. So here, like I said, we have the sampling step as well. The investigation is usually done by counting or extraction of yield components, for example, grape bunches, grape berries. Then we can also either weight them or just apply historical weight factors. And in the end, we extrapolate these numbers from the single plants to the whole vineyard. And this can be more or less successful. So here on the right side, you can see a table where we have the investigation of 4 different patches. And a vineyard manager tried to forecast the yield. Here in this column, we have the forecast, and this is the actual yield at the end of the season. As you can see, sometimes this works really nicely, and sometimes we have a large difference. So the mean yield estimation error is about 30%, which can be achieved by these manual sampling methods. And what is currently done in research is how can we estimate these phenotyping or yield estimation methods. And the idea is that we do not have a farmer running through the rows looking at single plants, but we want to equip a field phenotyping platform with some kinds of sensors, and every single plant can be monitored. And then we can, for example, for the yield, directly regress the yield. So we input some kind of information like an image, and then we actually get the yield in kilograms out of it. Or what most approaches do is that we have a preliminary step of counting the yield components or the berries, and then trying to correlate this with the expected yield. And also for counting, we have different methods which were already developed by different researchers. So we have some classical machine learning methods, for example, the radial symmetry transform, where we try to find round geometric objects in images. We could perform a density estimation, where we estimate these density maps, and the integral of these maps then give us the number of objects in the image. Or we can perform an instance segmentation. I will later go into detail what are the things behind these methods, but of course, all these methods also have some downsides. For example, these classical methods often have to do some assumptions. For example, for the radial symmetry transform, we have to assume the radius, which is dependent on the configuration between object and the camera calibration. For the density estimation, we have no detailed object characteristics, just the count. And instance segmentation approaches often suffer when they have to work with small objects, or if we have a two-stage approach, we also have to propose a number of objects. And for grapes, you can imagine we have a really, really large number here. So how does my thesis come into these problems? So like I said, the title is Contributions to Image-Based High-Throughput Phenotyping in Viticulture, and I contributed to 3 different topics in this part. So on the one hand, we have the part of detection and counting I already presented, and here I published 2 papers. On the other hand, this detection and counting is done as a preliminary step for yield estimation, where I also published 1 paper. And lastly, I also did some work on anomaly detection. But in the course of this talk here, I will only focus on these 2 pillars here, since otherwise it would be a little bit too long. So what is also good to know is that my research was done in the course of a project which was called Novisus. Novisus stands for Novel Viticulture Systems for Sustainable Production and Products, and the main goal of the project was the reduction of fungicide and pesticide use. I already explained why this is important and that we have different approaches how we can handle this. On the one hand, the option would be to breed fungal resistant varieties. This is done at the moment, but this takes a lot of time. So the breeding efforts and then introducing them to the market can take up to 20 to 30 years. So what was investigated in this project was the influence of different training systems. So the influence of management decisions on the plants and how they are resistant to different fungals. And here we investigated 2 distinct training systems. On the one hand, the vertical shoot position system on the left. This is the most common traditional system applied in Germany because it was developed for cooler climates. Here we have 1 perennial branch. The rest of the canopy regrows every year and is removed at the end of the season. The grape bunches are grown at the lower part of the canopy. They are very compact and the berries are homogeneous in size, but as you can see here, the canopy is rather slim and we do not have so much occlusion due to leaves. On the other hand, we have the semi-minimal pruned hedge. Here we have more perennial branches, as you can see here. The canopy is much thicker. We have smaller leaves, smaller grape bunches, which are also not so compact compared to the vertical shoot position system. This means these grapes are not as susceptible to fungal diseases, since the airflow between the berries is better. And what I tried to do now is I developed an image-based yield estimation pipeline where I counted the number of visible berries in images, and then I correlated this with the yield harvested in the field. And the approach has many single steps. I will quickly explain some of them and then some more in detail. So on the one hand, we have the image collection with the field phenotyping platform. On the other hand, we have to select relevant images. Then the counting in single images is corresponding to this first part of my thesis. And in the end, we also have to consider the vertical overlap. We have to count on plant level and not in single images. And in the end, actually estimate the yield. This corresponds to the second part of my thesis, and I will try to explain more in detail about these two parts. So the images were collected with the field phenotyping platform, which is called PhenoLiner. It's a grapevine harvester where the harvesting equipment was removed, and we installed a camera system inside. As you can see here, we have 3 overlapping cameras, which deliver 3 overlapping images showing the whole canopy of the plant. And since we drive along the row, we also have a horizontal overlap. And there are several methods how we could incorporate this. On the one hand, we could select images with a minimal overlap, but since we are interested into the yield estimation on the plant level, we decided to select images based on the plant positions, which we got from measurements of the trunk positions. So this is what we are now talking about throughout this talk. And the counting in images was realized with deep learning methods. So we have some kind of input, then we have some magic happening in the neural network, and in the end, we have an output. And there are actually several options how we could count in single images. I already explained some in my introduction, but now let's dive a little bit deeper into how we can actually count with deep learning methods. So on the one hand, we could perform a detection. A detection means we input an image and we get bounding boxes. Another option, or not so much an option, but we come to this later, would be a semantic segmentation. Here the output is an image mask where we have labels for the object and the not object. On the first look, it doesn't look like we can count with it, but I will show you that we can. Lastly, we can do the density estimation, where we have these density maps, where we only get the count of the objects in the end. And also we could perform an instant segmentation, where we have a mask of each single object, where we can actually distinguish between the single instances of the same class. And these different types of counting can be realized with different network types, because we also have different outputs. So let's start with object detection, which can be handled with a rather easy network. So we have an input image here. We have an encoder. Encoder are responsible for extracting high-level features from our image. And then we have 2 outputs, which are presented by a fully connected layer. On the one hand, we have this class label, which is given by one part of the network, and the other part delivers the bounding box. So if we see here, we can see we have these boxes, and each box then has a label saying this is a berry. But what we do not have is detailed information about the object size. So if we have elliptical berries, this cannot be nicely represented by this approach. On the other hand, for the semantic segmentation, we still have an encoder part where we extract features, but we also have now a decoder part, because we want to have an output in the same dimension as the input. So as you can see here, this decoder is responsible for reconstructing the mask and to go from this very high level feature representation, which was developed by the encoder, back to an image-based information. And here, like I already said, the output is just one label per pixel, and we cannot distinguish between the single berries. the same network type can also be used for density estimation, because here we also have the same kind of mask. So we have a mask in the same dimension as the input image, but here we don't have this class label. So we don't have just a label for each pixel, but here we have these density map peaks. And if we the label is not just 0 or 1 for berry or background, but here we have continuous numbers. And if we integrate over this whole mask, we get the object count. And lastly, we have an instance segmentation networks, which are very complex. So here the output is also more complex. We have a mask for each object, we have a bounding box and a class label. So here we need more parameters to actually get the output we desire. And what I proposed in my research is a new instance segmentation approach based on edges. So we are using the more simple network, which can perform a semantic segmentation, and we introduce an additional class called edge. So here we can actually distinguish between the same instances of the same class without using this very complex network structure from the instance segmentation, and at the same time leveraging the high-level information which we could get from the instance segmentation approaches. So now we can count, but we have to keep some challenges in mind. So what I wanted to investigate is 2 different training systems. Here we see example for these 2 different training systems and 2 different varieties, and our approach should work on multiple training systems and multiple varieties. So that means we have different berry colors. Felicia, for example, is much more shiny compared to Riesling. And as you can see here, the berries are very compact, the size is homogeneous. And on the other hand, for the semi-minimal pruned hedge, we have a varying berry sizes. There is not this compact structure, but nonetheless, our approach can handle both cases and both varieties. And what we did is we used the network presented here. So on the one hand, we have a very lightweight encoder with only with not so many parameters, which is able to extract a very dense feature representation. And we used the DeepLab version 3 because this network is known for refining the segmentation at edges. And as you can imagine, since we are actually introducing a class edge, this is very helpful. And now to show you some results, what you can see here is on the x-axis, the actual number of berries which was manually counted in the images. And on the y-axis, we have the predicted number of berries. And this is a correlation plot. So if the actual number of berries, which is our reference, would be 100% correlated with the predicted number of berries, we would have all the points here on this dashed line. And the actual situation can be seen here in these red dots. And the correlation line that is actually happening is here, the black line. And as you can see here, we have in all cases a very high correlation between the actual number of berries and the predicted number of berries of up to 95%, which is very convincing and is a good preliminary step or result for the step of counting for yield estimation. If we put this now into perspective with the other presented options, so we trained 2 other networks. On the one hand, the mask RCNN, which is an instance segmentation network, and a network for the density estimation. And we showed that our network has more convincing results. But now we have to say that there are actually reasons why the mask RCNN underperforms, because we have a limited data set, but we have a very large and complex network, which cannot be sufficiently trained with the limited data set that we have. And since we have so many parameters, also the inference time is very low. So we are talking about hours for the mask RCNN and minutes for the density estimation or our approach. At the same time, we are also able to deliver additional information, for example, the object size, which cannot be delivered by the density estimation. So now I showed you how we can detect and count single berries in images. And now I want to tell you how we actually come to the yield estimation. And the first thing we have to talk about is how do we handle the vertical overlap. And how we do this is by using the masks we developed in the counting step. As you can see here, we discarded all of the unnecessary background information. This is the image from the middle camera. This is the image from the lower camera. We extract patches around regions of interest, in this case, grape bunches. We restrict the search space based on the camera configurations. So we know the cameras are aligned vertically, and we have calibration parameters. And then we correlate this patch with this search space, and then we find the matching image patches and can account for objects which are present in two images. And now, if we have the number of visible berries per plant and we have the reference yield, we can investigate how these two numbers are correlated. Here we have this for the vertical shoot position system and for the semi-minimal pruned hedge. But let's start with the left side. So here we see that we can identify clusters for different yields where we collect the data. So in 2019, we had less visible berries, and we also had a lower yield. The cluster in 2018 is nicely visible in the middle, and here we have 2020. On the other hand, as you can see on the right side, there is no clear trend apparent for the semi-minimal pruned hedge. So we can have the same number of berries here. So everything on this 400 means we see 400 berries on this plant, but the yield can highly vary between 3 kilo up to 7 kilos. So why is this the case, and why do we have problems with this? For this, let's also look at other people who applied or who investigated yield estimation. So these are images of these data sets, and as you can see, the visibility of the yield components is very nice. On the other hand, these are our data. And as you can imagine, here, the large amount of leaves, which is usual for the German farming practices, makes it hard to actually see all the berries. So here you can see in the yellow images of the plant before defoliation. And here we investigated how much berries would actually be there if we removed the leaves. And as you can see, a large part of the berries are actually occluded by leaves, which accounts for this large deviation between the number of visible berries and the reference yield, especially for the semi-minimal pruned hedge. If we translate this for numbers for up to 30 plants where we investigated this, in yellow here you can see the number of visible berries before defoliation, and in purple you see the number of berries after defoliation. And here you can see roughly that we have double or 3 times the berries after defoliation, but this is really severe for the semi-minimal pruned hedge, where actually only a small part of the berries is seen before the defoliation. If we translate this into percentages, you can see for the vertical shoot position system, this is up to 300%, but for the semi-minimal pruned hedge, we have up to 2000% of occluded berries. And down here, we see histograms of these plots, and you can also see that there is no clear distribution. So we cannot just estimate one factor which can be applied to all plants, since the deviation of the leaf occlusion is very large. Nonetheless, we attempted a yield estimation for the vertical shoot position systems, because we were still able to handle this. And if we just naively multiply the number of visible berries with a weight factor, you can see that we underestimate the yield, which makes sense since there are a lot of berries we do not see. If we just naively double the number of berries, we overestimate the yield. And if we use a leaf occlusion factor of 70%, which is the mean leaf occlusion we determined by the experiments I showed before, we can actually have a mean average error for the yield estimation of 27%, which is still on par with the industrial standard without the manual sampling. So let me now summarize what I investigated in my thesis throughout the years. So on the one hand, I developed a new instance segmentation approach via semantic segmentation by introducing edge information. Then I contributed by investigating yield estimations under realistic conditions. And also I identified the limiting factors with an extensive leaf occlusion experiment. And lastly, what I did not talk about is that I also contributed 2 papers on anomaly detection in grapevine via supervised classification and semi-supervised image generation. And now, what is also interesting is what would come next. So what one really important thing is to talk about platforms and sensors. So right now, most of my data were collected with this really big truck, which is this grapevine harvester. But this machinery is very heavy and it can only be used in relatively flat vineyards. So one future direction would be to develop smaller sensor platforms, but then we have less controlled image acquisition situations. So here the imaging equipment was inside the grapevine harvester. So we had an artificial background and artificial lighting. On the other hand, if we have this smaller phenotyping platform, we have sunlight, we have shadows. So here we would actually need to make our networks more robust or to be able to adapt to different domains, for example, different weather conditions, different vineyards, etc. And then, especially for the yield estimation, we would need to handle the unseen. So one initial work was actually done by Jana Kierdorf, where she trained a generative adversarial network, which estimated the number of berries behind the leaves based on the leaf defoliation dataset I presented already. Or if we are able to generate what is behind the leaves, we could also think about directly regress the yield from the images without actually doing the counting. This is less interpretable, but would also be a very interesting future research direction. And now to summarize this in my thesis. So I already showed you that I provided 5 publications. I talked about these 3. The other 2 are the publications about the anomaly detection, which I did not talk about today. And I also wrote some other publications and collaborated with other people, not only in grapevine, but also for soybean or ear counting, and so on. So with this, I want to thank you for your attention, and I hope you got a little insight into what I did throughout my whole PhD.